I welcome you back to another class from the Nutrition in Humans. And for the previous classes where we have looked on toward the structures of the alimentary canal. So in today's class, we will see that how come the physical and the chemical digestion they are being attained here in the case of the alimentary canal. So as you could simply see by the physical digestion, what we mean is that some physical processes would be involved. Like we would not use any of the chemical, but some of the physical processes. And the role of the physical digestion is that we have to physically break down the large food particles into the small particles, and in that way, their surface area to the volume ratio would be increased. And that would make it easier for the enzymes to be digesting them more into the small constituent particles. So the situation is that where come this physical digestion would be taking place. So you need to remember the parts of the alimentary canal where this would be accomplished. So we have the mouth. After the mouth, we have this stomach with us. And after the stomach, we have got a situation of the physical breakdown that we call this as. So this would be under the impact of the bile. So you know that in the mouth we have got the incisors, canine, premolars, and the molar teeth, and they would be cutting the food, they would be grinding and crushing the food. So you see, physical processes are being involved. Now, when we talk about the stomach, so the situation with the stomach is that the wall of the stomach is highly muscular. The stomach not only be doing the chemical digestion, but the physical digestion is that when you people eat up the food, so you literally feel some sound coming out of your stomach. So the situation is that the stomach has the muscularity in it or the muscles in it, and those muscles, when they try to be pushing on toward the food, so that would be equal to the physical digestion. Now, when we talk about the bile, so bile is a juice, but why we say this is not bringing about the chemical digestion? Because this bile does not have any of the enzymes in it, but rather than this has got, you could say, some pigments in it, bile salts and the bile pigments in it, and that would do the emulsification of the fat, where the large fat molecules that we have with us, so they would be broken down into some small fat droplets without the use of enzymes. So for the mouth, stomach, and the bile, you have to be remembering this physical digestion. Now, moving onward, we have to see the chemical digestion, and the chemical digestion simply means that we would have some digestive juices, and in the digestive juices or the chemical secretions, we have the enzymes, and these enzymes hydrolytically, in the presence of water, would break down the food particles into the smaller ones. Now, you would not be able to understand this chemical digestion until or unless you recall for a while that what biological molecules were. So what we are actually eating in the daily life is we are eating the carbohydrate, and among them we are eating monosaccharide. Like we eat glucose, galactose, and fructose. So they are already the simple sugar, so they would not require any further digestion. But when we eat double sugars, like when we eat the maltose, the lactose, and the sucrose, so since they are the double sugars, they are the disaccharides, so they would need the digestion. Likewise, we are eating complex sugars, like we eat the potatoes and the rice, and they have the starch in them. So they require the enzymes to be breaking them down. Now, how the maltose was formed? I combined glucose and glucose to be making maltose. The lactose was formed by combining the glucose with the galactose. The sucrose was made by combining the glucose with the galactose. So when they would be broken down, so we would get the glucose and glucose from maltose, the glucose and galactose from the lactose, and the glucose and the fructose from the sucrose. And from the starch again, we will get the glucose. Now, the proteins that we are eating, like if I'm eating the chicken or if I'm eating the mutton or the beef, so the protein that we eat, it has amino acid and the polypeptide combined together and we make the protein. So if I would eat it up, so I would revert back to the polypeptide chains and to the amino acids. The fats that we eat, they are made up of three fatty acids combined with one glycerol. So when I would eat these fats up, so again I would break these fats back into the fatty acids and the glycerol. Now for the each part of the elementary canal, we must know that where any of the biological molecule would be digested. So here I have the biological molecule that I'm eating, and here we have the site of their digestion. So if we are eating any of the sugars, so in the mouth there would be only the partial digestion of the starch. The maltose, lactose, and the sucrose, they would not be digested in the mouth. In the stomach, there would be no digestion of any of the sugar because we have no enzymes for digesting them. In the small intestine, we would bring about the complete digestion of the sugars. Now, when we try to talk about the proteins in the mouth, no proteins digestion take place as we don't have the enzymes for them. In the stomach, the proteins they would be partially digested, not completely, but part of the protein would be digested, and then again in the small intestine, we would bring about the complete digestion of the proteins. And as far as these fats are to be bothered about, so in the mouth and the stomach, there would be no digestion of the fats, but in the small intestine, these fats again they would be completely digested. Now we will pick each and every biological molecule and we will see what's happening to them. So in our daily life, in the sugars, we eat starch, that is there in the potatoes, rice, and bread. We eat the maltose, that is there in the fruits. We take up the lactose sugar, that is in the milk, and when we eat the fruits, it has a sucrose in it. Now, we have to see the situation that what happens to these sugars or the carbohydrates in the mouth. So, in the mouth, when the food would be available, so the salivary gland that I have caught at the back of my lower jaw, so they would produce a saliva out of it. And the saliva would have an enzyme, salivary amylase in it. And this salivary amylase would only be able to break down the starch into maltose, partially broken down. Like some of the starch would be hydrolytically broken down into the maltose, but some starch would remain undigested here. Okay, and that leftover starch that we have going from the mouth to the small intestine, so it would be acted upon by the pancreatic amylases, but we will see here later. So this means that the sugar that are going from the mouth to the stomach into the small intestine, there would be maltose going, sucrose going, lactose going, and the leftover part of the starch going. So now here you see at the duodenum of the small intestine, the leftover starch has come, the lactose, sucrose, and the maltose has come. So now the pancreas would say that, oh, you have brought for me the leftover starch. So this pancreas would now release the pancreatic amylase here. Okay, and we will see later that how do the lactose, sucrose, and the maltose would be digested. So you see that the pancreas, they would release the pancreatic juice out of them. The pancreatic juice has a pancreatic amylase in it, and that pancreatic amylase would work on the leftover starch and hydrolytically would break down into the maltose. Now this means we are done with digesting the starch to maximum of maltose. Now we have to do something to the maltose, lactose, and sucrose. Earlier I have mentioned in the previous lecture that we have these villi with us, isn't it? And in the villi, we have got these cells, and these cells, they were the epithelial cells. So when we take one epithelial cell out of it, so in the cell membrane of the epithelial cells, now here we have the maltases, lactases, and the sucrases enzyme, and they are not the free enzyme, rather than they are immobile enzymes. Why? Because they are present with the cell membrane of the epithelial cells of the villi that we have here. So here something would happen to the maltose, lactose, and the sucrose. So in the epithelial cell, we have this maltase enzyme, we have the sucrase enzyme, and we have the lactase enzyme. The maltase enzyme would work on the maltose, and hydrolytically, it would be broken down into the glucose and glucose. If I talk about the sucrase, so the sucrase, as a name is sucrase, so it would work on the sucrose and hydrolytically it would be broken down into the glucose and fructose. When we talk about the lactase that I'm talking about, so the lactase would work on the lactose and in the presence of water, this would be broken down into the glucose and lactose. So here you see that out of all the sugar that we have eaten up, so at then we have got the simple sugars or the monosaccharide and they can directly enter into my blood capillaries and from there I would take them to the cells of my body. Now moving onward, here we have to be talking about the chemical digestion of the protein. In the mouth there would be no digestion of the protein, but in the stomach the protein would be partially digested. The situation that we have to create it, that in the stomach there is presence of the gastric glands. And these gastric glands when stimulated, so they would produce a juice out of it, and the name of the juice is gastric juice. And the situation with the gastric juice is that they have an inactivated enzyme in it, and the name of that enzyme is pepsinogen. Okay, so the first situation is that how are we required to be activating this pepsinogen? So on the pepsinogen, this hydrochloric acid would be working, and pepsinogen would break down into the pepsin. And when we have activated the pepsinogen into the pepsin, so now this pepsin would work on the proteins that we have got in the stomach, and in the presence of the water, they would break down into the polypeptide chains. But some of the proteins still would remain undigested in the stomach. Now, we have to completely break down the polypeptide chain and we have to do something to the proteins that remain undigested in the stomach. Now, moving onward, these proteins they would be brought into the small intestines. Now, when the protein has come into the small intestine, there would be the contribution by two of the organs, like the pancreas would come into the play, and the intestinal glands would come into the play. The pancreas, they would release the pancreatic juice, and the pancreatic juice has an inactivated enzyme in it, and that is trypsinogen. Intestinal gland, on the other way around, they would produce an intestinal juice. And that intestinal juice will have the erepsin enzyme and it would have the enterokinase enzyme. Now, the first scenario is that how would we be able to convert this inactivated trypsinogen to the trypsin? So the enterokinase enzyme that we have got being produced from the intestinal gland, so it would work on the trypsinogen and it would revert into the trypsin. Now, what the trypsin is going to do, that the protein that remain undigested in the stomach, they would work on it in the presence of water and we would make polypeptide chains out of it. Now, the polypeptide chains that we have made in the small intestine from the leftover protein, and those polypeptide chains that are coming from the stomach, so they would be acted upon by the erepsin enzyme in the presence of water. Now, we would make amino acids out of it. So here we are done with the complete chemical digestion of the protein. Now here we understand about the digestion of the fats. How the fats that we eat, they have three fatty acids and they have one glycerol in them. So none of the fat digestion happen in the mouth nor in the stomach because we have got no enzymes being produced for the fat digestion. So now the situation is that here in the small intestine, the fats would come here. And when the fats are coming here, so now there would be a contribution coming from the liver, and there would be some contribution coming from these pancreas that would assist in the digestion of the lipids or fats. Now, what the liver is going to do, that liver would produce on the inside of it a juice, and that juice is the bile juice, and it has the bile pigments in it. And for some period, short period of time, these bile juice and the pigment they would be stored here within the gallbladder. The pancreas, they would release the pancreatic juice, and that pancreatic juice would have the pancreatic lipases in it. And the liver from its gallbladder is releasing the bile juice here into the duodenum of the small intestines where we have brought the fats. Now, what we try to see here is, like the scheme is, that the liver has produced the bile juice. And this bile juice would emulsify the fat. And by the emulsification of the fat, we will see that the fats, when they are broken down into the small fatty droplets, so this is emulsification. And then the pancreas that we have got at this point, like here, so now they have produced the pancreatic juice, and that would have the pancreatic lipases in it. And these would work on the fat droplets that were made by the emulsification process. And in the presence of water, the pancreatic lipases would break down these fatty droplets into the fatty acid and glycerol, and in this way, the complete digestion of the fat would be accomplished. Now, if I were to tell you that how many are the major steps in the complete digestion of the fat, so one would be the emulsification, and the second would be the kind of the breakdown that we have to see. So this would be the chemical breakdown, and that chemical breakdown would be done by the enzymes, and these enzymes, they are the lipases. This is not the thing that the lipases alone cannot digest the fat, but their efficiency of digesting the fat would be less. But if we try to be emulsifying the fats with the help of the bile juices, then the catalytic breakdown by the lipases becomes easier. Now, what emulsification is, that when the bile which is coming from the liver, when it would break down the large fat molecules into small fatty droplets, so this is what we refer to this as emulsification. Now, the advantages of the emulsification are that this emulsification increases the surface area. Like this large fat molecule has lower surface area, these small fatty droplets, now their surface area has increased. The bile, they would also reduce the surface tension of these large fat molecules when they are converted into the small fatty droplets. Now, when these things in terms of an increased surface area and reduced surface tension is attained for these small fatty droplets, now these would be worked upon by the lipases enzymes, and these lipases enzymes, when they would work on toward the fatty acid droplets, and ultimately in the presence of water, they would break them down into the fatty acids and the glycerol. So here we are done with understanding that how come the complete digestion of the fat would be brought about by. I hope you would be clear with it, and in the next class we will look on toward the absorption phenomena.